Hello everyone, I am Dr. Ponchitra, Assistant Professor, Department of Physics, Sadakatullah Appa College, Tirunelveli. Today we will discuss about scanning electron microscope, working principle, advantages and its applications. The scanning electron microscope is an improved model of electron microscope. It is used to study the three-dimensional images of the samples. SIM plays an important role in various fields like material science, biology and nanotechnology. Let's see in detail. Anyone guess this picture? Yes, this is the three-dimensional SIM image of silicate, silver nanoparticles and gold nanoparticles. The contents of the session are introduction, principle, construction, working, advantages, disadvantages and applications. The scanning electron microscope is an enhanced version of the traditional electron microscope. SIM is used to study the three-dimensional images of the specimen. History of SIM. The development of SIM occurred over several decades. In 1935, Maxwell developed the first scanning electron microscope. In 1942, Manfred built the first true SIM. In 1965, Charles developed the first commercial SIM. Principle of SIM. When the accelerated primary electrons strike the sample, they generate the secondary electrons. These secondary electrons are collected by a positively charged detector which helps create a three-dimensional image of the sample surface. This is the image of scanning electron microscope. First one is electron source, second one is condenser lens, aperture, scanning coil, objective lens, backscattered electron detector, the samples, detector, scan generator and amplifier is connected with computer. Construction of SIM. The main parts of SIM include electron source. It produces the primary electrons. Condenser lenses, that is C1 and C2, it focuses the electron beam. First condenser C1, it reduces the beam diameter. Second condenser C2 is used for fine-tune beam size. Aperture, a metallic disc, that small central hole that restricts the electron beam and eliminating off-axis electrons. There are two types of aperture. Condenser aperture, it controls the beam current. Objective aperture, it reduces the spherical aberration. Scanning coil, these deflect the electron beam in raster pattern across the sample surface. It enables high resolution image formation pixel by pixel. Next one, objective lens. The lens is used for coverage of electron beam onto the sample surface. Backscattered electron detector. It detects the high energy electrons. Samples. The specimen being examined usually coated with the conductive material. For example, gold. Detector. It captures the signal generated by the electron sample interactions, that is secondary electrons, backscattered electrons and X-rays. Scan generator. It controls the scanning coils to deflect the electron beam in a precise raster pattern, that is XY over the sample. Output. It generates the timing signal required for the image construction. Next one is amplifier. It boosts the weak detector signal to improve signal to noise ratio. Then working principle. The stream of electrons are produced by the electron gun and these primary electrons are accelerated by the grid and anode. These accelerated primary electrons are made to be incident on the sample through condensing lens and scanning coil. These high-speed primary electrons on falling over the sample produces a low secondary electrons. The collection of secondary electrons are very difficult and hence a high voltage is applied to the collector. This collected electron produces scintillations on the photomultiplier tube and converted into electrical signals. These signals are amplified by the video amplifier and fed into the CRO. By similar procedures, the electron beam scan from left to right and the whole picture of the sample is obtained in the CRO screen. Advantages. SIM offers many advantages. High resolution, three-dimensional surface imaging, wide magnification, elemental analysis also possible. Disadvantages. However, SIM has some limitations. It requires vacuum condition. For sample preparation, non-conductive samples need metal coating. Cost and maintenance is expensive. Surface only imaging. It cannot analyze internal structure like transmission electron microscope. Applications. SIM is widely used across various disciplines. For material science, SIM is used to examine the microstructure, surface morphology, fracture and grain boundaries in metals, ceramics and polymers. For example, zinc oxide nanorods, nanoflowers, CDO thin films. In nanotechnology, it is used to analyze the nanoparticles, nanowires and nanolily flowers. Biology. SIM provides detailed image of biological samples such as stem cells, tissues, blood clot, bacteria and etc. I hope you understand the working principle, advantages and various applications of the scanning electron microscope. SIM is an essential tool in the field of material science, biology and nanotechnology. Thank you for watching and keep exploring the world of science.